All right, so good morning, everyone. Thank you for being here. Welcome online too. So this morning we are very lucky to have Chris Cassa come give our primer today. He's an assistant professor of medicine in the Division of Genetics at Brigham Women's. He's a lecturer at MIT and also an associate member at the Broad. His lab develops computational and experimental methods to determine which rare genetic variants really increase disease risk using large-scale clinical and genomic data, functional genomics, and machine learning. A key goal of his is to distinguish harmful variants from those commonly found in otherwise healthy people. So I'll take it away. Thanks.
Thank you so much. Thank you to the organizers and thank you for coming to MPG Primer. I'm really excited to be here and I welcome questions throughout, so please raise your hands, or if questions come into the QA, feel free to let me know. And so today, as you mentioned, I'll be talking about new approaches and data sources for coding variant interpretation. So this is me. And I want to point to two MPG primers that are related. So one was recent. It was in this fall. Pam Robertson talked about ClinGen, ClinVar, and other clinical genomic resources. And so this sort of gives the lay of the land of the resources for data that are available for gene and variant interpretation, and the working groups and the processes and the VSEPs, the variant curation expert panels that are working to curate a lot of variants that have multiple submissions from clinical labs and diagnostic labs. So this was a great foray about these resources. And if you wanted to learn more about ClinVar, she walks through an example of the variant in ClinVar and looks up all the available information. Then the other one was from 2024 by Chrissy Austin C. And that is all about the in-depth process of variant curation. So if you go to a clinical lab and you submit a sample because you have a rare disease in your family, she walks through the process of what happens during that variant curation, and also about gene curation in general, making sure that there's enough evidence about a gene to be confident that that gene has an association with a disease to then be able to work at the variant level. And so these are great primers to walk through. And so now to my side, I wanted to, many of you have probably seen this before, but the NIH had these NHGRI had these bold predictions for human genomics by 2030. And one of them, number 7, was the idea that we would be basically getting rid of all VUS, this idea of variants of uncertain significance. And the idea is that every single time we have a variant, we should be able to know what its clinical relevance is so that we can tell a patient and use it in genomic medicine. And the reason why these VUSs are so tricky is that human genetic variation is often very rare. And there are so many variants out there, maybe 1 billion variants that we know about. And so the ability to curate these variants with clinical knowledge and experimental resources and things like that are vastly outpaced by the number that we continue to encounter. And so Sean Fayer had this paper in 2021 that just sort of showed that this arc is just incredible. We're highlighting and finding so many variants in the context of patients and biobanks, but we don't know what their clinical significance is yet. And so as sequencing expands, more rare variants are encountered, but there's often really limited evidence to decide whether or not those variants cause disease. And so this really matters if you're a patient. If you have a rare variant in BRCA1 or BRCA2, you might be really concerned about it, right? Especially if it's ultra rare, it's a non-founder mutation, and you want to think about what's the clinical impact that might come from this. This is not one of the ones we know causes disease. It's not one of the ones that we know doesn't cause disease, or doesn't cause, is an extremely highly penetrant, or have a really severe outcome. So it might be somewhere in the middle. It might be something we've never seen and has the same high risk or the low risk. So it's really concerning. And so the ACMG has this list of secondary findings genes that are just so clinically actionable that you want to know what is going on with them if you have a variant that is pathogenic or likely pathogenic in them. But what we have to do is get these VUSs, these rare variants that are in these genes, classified as either benign or pathogenic so that they can be used to counsel patients, not only for their families, but also for their own clinical care. And I just want to put a little scope on this. So if you look in, if you look in the UK Biobank, about 1 in 20 people has a rare non-synonymous variant in BRCA1 or BRCA2. And if you look at, I should have called these out better. these 9 genes, these are sort of known as the CDC tier 1, maybe even useful for population screening. These are like familial hypercholesterolemia, LDLR, PCSK9, ApoB, Lynch syndrome, colorectal cancer, thinking about the 4 genes that are commonly associated with mismatch repair, and these hereditary breast and ovarian cancer syndrome genes, BRCA1 and BRCA2. 1 in 6 people carry a rare non-synonymous variant in one of those genes. So the number of people that carry some rare variant in these genes is really high in comparison with the number that actually have been classified as pathogenic or likely pathogenic. And that's a relief, because most of us are not walking around with a severe, highly penetrant variant, hopefully. But at the same time, we have this big unmet classification need. And so many patients still do have genetic risk that don't learn about it right now because we aren't certain enough about those variants. And that's what we want to work on today. And so we have this need to accelerate variant classification, because variant classification is determining whether or not there's sufficient evidence for using this information about a variant that you encounter in a patient in clinical practice. And the process looks something like taking a DNA variant and classifying whether it's pathogenic, benign, or uncertain. And the classification happens once some amount of evidence crosses a bar. In the case of being pathogenic, it has to cross this bar of a certain amount of evidence points, and we'll go through that a little bit. And if it's going to be called benign, it has to go through this bar and go into sort of the negative territory of negative evidence in terms of the number of points that are available for it. And so what we're thinking about when we're doing this process is thinking about aggregating a bunch of different types of evidence and trying to see in totality, does that evidence sum up to being likely pathogenic or likely benign, or are we still uncertain, or in this VUS category in the middle? And to do this, you might want to ask, how do we do this process to determine if a variant is pathogenic? So we know that there's going to be some evidence. I wanted to call out first, there was this sort of earlier era of variant classification. Why did this get so complicated? Back in the day, maybe 20 years ago, if a rare variant was encountered in a patient with a strong family history in a known gene, like you found a patient with a strong family history of breast cancer, and they have a variant that's a non-coding variant, a coding variant, a non-synonymous coding variant that's rare in BRCA1, a lot of those variants got classified as likely pathogenic or pathogenic by the clinical labs at the time because we hadn't standardized, and that resulted in a lot of false positives. And so what we have now, about 10 years ago, there's this big push to standardize, and Sue Richards and Heidi Ream from the Broad pushed this platform forward called the ACMG-AMP Sequence Variant Interpretation Guidelines. I'll call these the SVI guidelines. And what they did was they standardized various evidence types that you might use to decide whether a variant is likely to be pathogenic or likely to be benign, and they calibrated these associated strength levels. Given the type of evidence, how likely was it to be truly causal or truly non-causal? How much did it help you decide whether the variant actually causes disease? And then there have been various evolutions of this. So as you can imagine, over the last 10 years, we've learned a lot, and new data sources have been developed that have kind of taken one of these guidelines at a time or 2 of these guidelines at a time and tweaked them and tried to understand how strong the evidence is, whether it is actually a little bit stronger or a little bit weaker than they originally thought 10 years ago. And there's also this Bayesian adaptation of this framework from 2018, which tries to kind of simplify and clarify the way that this evidence is weighted and puts it in a clinical framework that kind of combines this evidence together a little bit more cleanly. And there's an updated version forthcoming of this, and so there'll probably be a talk next year about the version 4.0 of the SVA guidelines. Hopefully this will be coming this year. And the premise of these guidelines is that no single source of evidence is sufficient to classify a variant. Even if you have a really high quality functional screening assay where you have a wonderful model system, there's still a lot of noise, experimental noise, or even the question that the platform might only work for certain variants in the gene and only may be a good proxy for recapitulating a functional human phenotype within the cell system. It only might work for certain variants or certain genes, and it's unpredictable whether or not it will work. So that, even though it's excellent evidence in aggregate, at the individual variant level, it's not sufficient enough to call this variant a clinically pathogenic variant. And the same thing with computational predictors. You can imagine they're quite good now, but they're still imperfect. It's not enough to say, oh, alpha missense says it's pathogenic, so that we better counsel the patient. That's not enough. And even a very strong family co-segregation with disease for a variant is on its own. Like a LOD score is not enough in one family to be able to call a variant as likely pathogenic. So for rare variants, there are ways to call it benign, but no single source is enough to call it pathogenic for a rare variant. And so what we do is this kitchen sink approach. So we try and take every piece of evidence we can develop about a variant. We try and take all the clinical case evidence that we might have. And what does that look like? It's like a whole bunch of submissions from clinical diagnostic labs where that variant has been seen in a patient or in a family. So they take all the probands and they think about all the segregation within families of that variant with disease. We think about functional screening data if it's available from a MAVE or deep mutational scanning or saturation mutagenesis profiling assay. We think about computational predictions. We'll talk about that today, about the newer kind of next generation computational predictors. We'll think about population frequency data from resources like Nomad or from biobanks. And we can also think about contextual diagnostic data. Are there variants nearby this variant that you're thinking about that suggest that this variant might be more likely to cause disease or less likely to cause disease? And so we take this kitchen sink and we kind of put it all together into variant classification. And so, how does this look in practice? First, we calculate each form of evidence. That might be looking up. Sometimes that's manual curation for a person in a clinician in the lab has to actually go through the papers or go through the different lab submissions that are available for a given variant. Maybe this variant's been seen in 3 different families, in 3 different international clinical diagnostic labs. So they have to think about what forms of evidence are available, look up if there's a MAVE and try and see what the functional score was, where did it fall in the spectrum of functional scores? And then ultimately they have to calibrate the strength of each form of evidence, and then they have to combine it. They kind of have to sum it all together to think in totality, is this likely to be a likely pathogenic variant or a likely benign variant? And so I kind of mentioned this idea that the 2015 guidelines specified individual levels of evidence, strength levels that go with each of these forms of evidence. So if you had a population risk of a variant, let's say you encountered a variant in Nomad, and it was only in a couple of controls, but you saw it in a few patients, that might count as some form of evidence. Maybe it's +1 point of evidence. And maybe the computational tools don't agree or they think it's benign. So maybe that's -1 point of evidence. Oh, but a functional screening assay said it was damaging. So it reduced function of the protein when it was working. Or instead of working, it's not working. So that might be +4 points of evidence in favor of pathogenicity. And then so you sum these all together. Some of these forms of evidence won't be available for a given variant. Maybe there's no MAVE or something, or maybe there's no family case data. In totality, you sum these up, and then you get to some point in this number line. And if it kind of exceeds 6, it's likely pathogenic or higher, or if it's below 0, it's likely benign or benign. And so kind of putting this all together, this is how we decide whether a variant right now is likely to be pathogenic. Of course, this is different than the interpretation of this variant in the context of a patient. Any given patient may have specific characteristics like demographics or other risk factors, clinical risk factors, polygenic risk, or other things that can be combined or made to better assess what that patient's individual level of risk is. This is much more just about the variant itself, whether the variant causes disease. And so, good, I'm good on time so far. All right. So what I'm going to focus on today are 3 different core pieces of new evidence that are being developed that are powerful enough to kind of help us get a bunch of these variants of uncertain significance classified. The first we'll think about are next generation computational predictions. So these are things that are using structural models, alpha missense, alpha genome, foundational models, trying to think about how do we better use that evidence and the strength of evidence from those computational predictors. And the second part is a project I just worked on, which was thinking about how do we extract information at the variant level about patient disease risk from large population cohorts. What can we learn from biobanks about the risk of a variant causing disease, how many patients we might have seen it in, whether they have disease or not, And then the final thing, I'll think about a little bit of some of these new functional screening platforms that are really high throughput, that are pooled, or I don't know, from CRISPR-based. So we'll think about those three forms, not only on the new sources of evidence, but also on the calibration of evidence. I want to take a second to talk first. Are there any questions about the framework itself and how we kind of go about integrating the evidence that's available? Great. Okay. I'm not sure how many people had seen it, so I wanted to kind of make sure everyone had a view of it. So the first section I want to talk about is about computational evidence. So I wanted to call out first, this is a lot of work from Biobank-derived, but also from the ClinGen Clinical Sequencing Working Group, the Computational Working Group. And it involves a lot of work from folks who are on that working group, including Anne O'Donnell from here, Peja Rajovic from Northeastern, as well as folks from Utah and from the NIH. And so when you think about new computational methods, these next generation models are, how many people have seen these sort of structural aware models like AlphaMissense or Primate AI 3D, which is both structure and evolution aware? These are sort of a leap ahead in terms of the quality of predictions and the accuracy of predictions that come out of these models compared to the older versions, which had much less data, right? They had, essentially, these are the sort of deep learning versions of computational predictions that we hadn't had before. Really understanding the structure, the sequence context, the deep evolutionary context of variants. And what I wanted to kind of call out about these is there are now some models, these foundation models and generative models, that actually can make predictions about every single variant in the genome. So they can make predictions about the impact of non-coding variants or how likely or unlikely these variants are to occur or to fix within species. And so these computational methods are considerably better than the prior methods. Still, they have that same caveat, right? Every individual variant may not actually be accurate, but in aggregate, they're high quality, right? So what we want to think about is, are there ways to take advantage of this knowledge that they are really high quality with really high scores to be able to give them a little bit more credit than they were originally given 10 years ago? Because 10 years ago, the computational predictions were good, but they weren't great. And so back, when these 2015 sequence variant interpretation guidelines were made for how to decide if a variant is pathogenic, they only gave supporting level of strength to computational predictions. So if multiple lines of computational evidence existed and they said that the variant is likely to be pathogenic or to impact function, that was given a supporting strength. That's only a little bit of credit in the sequence variant interpretation guidelines. So it doesn't get you over the hurdle, even with another form of evidence. So what we want to do today is think about, are there ways to calibrate this strength of evidence and see if there's a really high functional score on a really high quality computational predictive tool? Does it give us even more confidence that this variant is likely to be pathogenic? This is super nerdy, right? So like, we're trying to think about how do we get out of this binary classification scheme for computational classifiers. We might think of a variant being called pathogenic or benign by these classifiers, but most of them actually output the confidence that the variant is likely to be pathogenic or benign on a spectrum. Often it's between 0 and 1, or negative infinity to infinity. But you have these scores that come out on a continuous spectrum, and the goal is to think about, if you're really on this high zone of confidence that this variant is pathogenic from the predictive tool, and the predictive tool is really good, can you give it moderate or strong or even very strong evidence strength? And so what Vikas Pejavar and that computational working group did was they worked through calibrating whether or not a variant score from one of these computational predictive tools was good enough to be counted as a higher level or stronger level of evidence. And the way they did that was, you can take a score and say that score goes between 0 and 1. If you score every single variant that you know about from ClinVar, like take all these clinically certain variants that are either known to be pathogenic or benign, if you look at them, you kind of put them all on a number line between 0 and 1, you score every single variant, and most of the variants that are red appear to have high scores, and most of the variants that are blue appear to have low scores with this computational predictive tool. So let's say this is AlphaMissense, for example, right? So we're giving a score for every single one of these variants, and we're looking up in AlphaMissense what its score is, We're putting it on this number line, and we're just coloring it with whether it's pathogenic or benign. So the first thing we see right away is that the score is a pretty good discriminative tool. It's actually telling us that most of the variants that we know already are likely to be pathogenic do have high scores, and most of the variants that we know are likely to be benign have low scores. And the premise of this computational calibration is to just check it on time. Okay, great. The premise of this is that we want to see what is the score threshold by which we're almost so certain. Like if you drew the line up here and you said every score above 0.95 or something, how likely is it that that variant who's found with that score is likely to be pathogenic? How much more confidence? What's our positive likelihood ratio of actually believing that that variant is likely to cause disease? Given all the variants we know about that cause disease or don't cause disease, we've labeled them. How much more likely is it that a variant with this score in this range is likely to be causing disease? And what they did was they sort of calibrated this by calculating the posterior probability of whether, if you're in this score range, how likely you are to definitely cause disease. Like this would be very strong evidence, moderate evidence, or supporting evidence. And you can do the same thing in the other direction. You can say, if you have a really low score, how certain are we that that variant is likely to be benign? So you look at this score, if you're down in this range, almost every single point is blue, and that means that almost every single variant you find in that score range is unlikely to cause disease. And what they were able to do was kind of scope out these different score thresholds that basically say between 0 and 1, how likely are you to, if you're in this range, how likely are you to be providing strong evidence or very strong evidence or moderate evidence of causing disease? And the caveat that goes with this is you don't always reach these thresholds, right? You might actually have a score that's not as good as this at kind of, this is a really nice spectrum where you see like there is some range where most of the points are red and most of the points are blue, but you do have this middle range here where you're actually not that certain, right? If I told you, oh, actually the score is 0.5, how confident are you that it's a pathogenic variant? You shouldn't be very confident at all. It's probably about 50% confident, right? So that would be in that side of kind of uncertain range. So you don't have a lot of predictive value for deciding if a variant is damaging or disease causing in that range. So this is sort of thinking everything above here might be supporting, everything above here might be moderate strength, et cetera. Any questions on this? I know this is nerdy. I think this is the nerdiest thing that's going to be covered today. Well, I guess all a little bit nerdy. So yeah, exactly. Sorry, it's great. In depth. So I guess what I was going to point out here, this is a slide courtesy of Cassa. He shared these sort of wall of different scores. They went through each of these different computational predictive scores, and they thought about what is, he calculated essentially what is the actual score range that associates with strong evidence. And one thing I want to kind of point out, originally, this evidence was only counted as supporting, meaning it only had 1 point of evidence in the framework. But Cassa was able to calibrate at least 4 of these different computational predictors. These are kind of notable ones that are very commonly used in the field too, like Revel and Vest are used in the field very frequently. And he was able to find scores that were so high on these tools that almost every point was red above that range. Sort of, you go back to this slide before, he was finding like, oh, there are ranges of these scores where almost every point is correctly classified. And so we're pretty sure that there's actually very high quality evidence or strong evidence that this variant is disease causing from the computational evidence that's provided. And so that's kind of a big leap because this was originally counted as +1 or -1 point. Now you can count it as +2 or +4, depending on how high or how low that score is. And the same thing in the other direction. You even see Revel is even able to technically give very strong evidence, but they're not going to count it for that. Very strong evidence of benignity on the other side. And there are these same 4 scores are performing pretty well on both sides. There's also an updated paper that just came out last year, which goes through even some more recent scores like alpha missense and ESM1B and Varity. So this is just a big leap for this field to be able to count computational evidence as strong. And this is still getting grappled with by the sequence variant interpretation folks. people are starting to use it in practice, but it's not fully there yet. So I think this is really one of the things we're hoping is going to be really resolved by the upcoming guidelines, which are grappling with how to consider evidence from a function and impact of function. Of course.
Well, and they were like a similar sort of underlying function that kind of made them better, or was like there's similarity in that?
Oh, that's a great question. Yeah. So I should say they're all great methods, right? They're all taking different forms of evidence. So some of these are ensemble methods, right? So if you look at methods that pull a broad variety of different features, some are about evolution, some are about constraints, some are supervised. They're using label data, knowing whether something is causing disease or not causing disease. Those tended to be the methods that did the best. Of course, there are all these confounding factors that come from that, right? So like some of the variants that were classified using computational scores or functional scores. So there's always a little bit of circularity there, but this is a cool part of it that I think that, using more of the evidence and using the labels that have been developed by the clinical community have both been integral for that. But a lot of the newest methods don't use some of this, right? So they use, like alpha-missense is supervised, but it's using a much deeper breadth of sequence context and protein structural context, right? So this is sort of like kind of going even deeper into that world. Thank you. Great question.
While we're paused for questions, there was actually one more, a couple of slides back with the blue dots on the scale. Yes. So they have a question about the very high scores for pathogenicity. I understand there will be some error in this classification, but how do you deal with those blue dots that have very high scores?
That's a great question. So this is exactly, so the question is all about, hey, why are there some blue dots with really high scores here? And this could be correct. Like we don't know for sure if this computational score might be more correct than the clinical community. Maybe this was a really old classification done by one lab and that's all the information we have about it. Heidi and the ClinGen folks are doing a great job of cleaning those up. The team here as well at the Broad. At cleaning these up and thinking about, is there really that strength of evidence to call this blue or not? But what this really does is it thinks about this as a framework that every single piece of evidence, like a computational predictive tool, might not be perfect. So there might just be a range at which, this is actually pretty predictive. It's providing us maybe moderate level of evidence. There's some preponderance of red points, more so than blue points here. So that is some form of evidence, but it's not definitive form of evidence. This is sort of it. Oh, I guess I know it's hard to answer that online, but I think that the goal here is to say we acknowledge that either the classification itself might be wrong or the tool might be wrong. And either way, we're just still happy to draw some benefit of knowing that more of those variants are likely to be classified correctly in that score range. Cool. All right, great. Any other questions? Cool. All right. So I just want to cap off this section by saying, calibrating predictors provides the stronger level of evidence. And that's now almost on par with functional assays. So like actually doing an experiment in a lab, trying to see if that cell with a variant installed in it is likely to recapitulate a human phenotype. These computational tools are almost on par with that level of evidence from an experiment, which is really great. And notably, as I kind of pointed out before, these scores are available for every single variant. So it takes a lot of resources to perform a functional assay, but you can just run these tools now for every single variant. So you actually have a score and some amount of evidence for every single variant in the genome. It's really a powerful thing that we're able to kind of give that a little bit more credit. And another thing to kind of point out is that single tools are better than an uncalibrated consensus approach. So one of the things we saw, given the original guidelines were written about whether there was a consensus among tools, one of the things here is we're sort of focusing now just on the score and the strength of evidence from one tool at a time. And that makes sense because I think kind of going back to your question, a lot of these tools are using a lot of different forms of evidence already. So they're not, you don't really need the same consensus among different types of tools that are drawing from different types of evidence because these ensemble methods or these high quality deep learning methods are now drawing from a lot of different sources of evidence, both evolutionarily as well as functionally. All right, so that kind of caps off the.
Sorry, can I ask a question?
Absolutely.
So I think in the Petrover paper, or like just traditionally, people say, you can only use one computational tool, like only one counts. Now that we have a better idea of where the data is coming from for different tools, do you think there will be changes to that rule?
That's a great question. So the sort of question is, make sure I'm getting it right, but like you might be tempted if this score comes out, let's say both Revel and MeetPred2 both have really high scores. Am I more confident that this variant is disease causing? And then the question is this double counting, right? Both of these are drawing from very similar forms of evidence. So, okay, you probably want to be careful about that. But we haven't really gone through what does it mean to have a new form of evidence and how much more does this count or not count? And I think it's probably a tricky territory. In fact, it looks like the next sequence guidelines may be going in the other direction where they're going to count impact of function as one whole class of information and evidence and maybe even cap that. So they're sort of talking about we don't want to give too much credence to any one form of evidence because we know on the whole, it might be individually correct, but on the whole, it's still risky, like that you could make, misclassify or make a false positive classification. So I think that maybe to answer it better to say, I think it's probably fraught, right? So you don't want to, it's just tricky to know whether or not those forms of evidence are truly distinct, whether the labels that were used to train the ones that are supervised, for example, whether those are sort of giving a lot of information to the same type of deep learning model that could learn it in some way that you didn't anticipate, even though you use different features to put into the model. There's some risk about that. I think it's a great question though. Like, can you do it? Is probably, but like, not yet because we don't know. We haven't kind of gone through it yet carefully enough. Any other questions? Yeah.
How do you choose what tool to use then?
That's a really good question. So I think this is one of the things that clinical labs really grappled with, especially in the US where we have like a regulatory environment where we're thinking about like, what is the right answer? What's sufficient for a consensus, right? Now I think the answer is still probably different labs are using different tools. The ClinGen community will probably get there to kind of make recommendations, but I think this is a kind of good way to quantitatively do it. So the information's out there now. People are still adapting to it and thinking about adopting it, but I don't think there are specific tool recommendations yet.
So would you run all of them and then choose, or would you pre-choose and then run that one tool?
Great question. Yeah. So I think you should pre-choose and then run that one tool.
Okay.
And then, but I mean, I'm sure really not positive actually how that's done in practice right now, but I think that's more sound, right? This way you're not kind of cherry picking, oh, this one tool said it's bad.
Yeah.
And I think that's one of the things that's been challenging is that any one tool has some false positives. And so you just have this higher chance of false positives. Still within the framework, this is counted as some form of evidence. It's not enough to get you over the hurdle on its own. So this is sort of why the framework is so protective around that. Great question. Any others? Okay, good. So I wanted to jump to the next section. This is some work from my lab. So I wanted to kind of call out Vanille and Tian, but also joint work with folks from the computational clinical working groups from ClinGen. And the goal of this project was to see, now that we have such big biobanks, we use the UK biobank, for example, 500,000 people. We wanted to see, is it possible to look at a variant and decide whether the patients who have that variant in the UK biobank or in another biobank, like all of us, whether they are likely to have disease or not. And this is another form of evidence that we haven't really had before at this scale. The way that this evidence was used in the original 2015 framework was that there was this idea that if you had a highly penetrant severe disorder, you might find a few cases of variants, maybe a couple of probands or a couple of families around the world that had this variant, and they also have the disease. And that might give you some more evidence or some more certainty that this variant is likely to cause this disease, just because there are multiple people out in the world in the wild with what should be a bad variant. It's a severe disease, so you shouldn't expect to see it in too many people, this variant, and if it's highly penetrant. And so the way that this worked is if you kind of counted cases traditionally. And so you would look and you see 2 patients who are in different families with the variant. Maybe you count it as supporting evidence, or 6 patients, it might be moderate evidence, or 10 patients. It could be strong evidence. This is tricky because there's sort of no question about how the ascertainment was done. So if you're looking at breast cancer, so many more people have been sequenced and screened for breast cancer than for another rare Mendelian or monogenic syndrome, right? So you have a lot of confounding about how those patients were ascertained, who were sequenced, what that data kind of, how likely is it to be predictive? It might differ. And so the various variant curation expert panels from ClinVar for different disease areas and for different genes have actually counted this evidence differently, depending on how many cases were there. The sequence variant interpretation guidelines also allow for this idea that if you have a population cohort or a case control cohort, you can look for an odds ratio where that variant has an odds ratio of 5 or greater of causing disease. So you're 5-fold more likely or higher to have disease than not having disease if you have this variant. And if you look at a variant and it has an odds ratio of greater than 5 and it's above 1 for the lowest confidence interval, the lower bound of the confidence interval, you can count that as strong evidence. What does that really mean in practice? We go to a biobank, we look at the variant. Do more people with this variant have disease? 5-fold more people have disease with this variant. And so we asked, can biobanks provide this evidence of variant enrichment with disease? Usually what you would want to do is use a case control cohort to do this type of calculation. So we were asking the question, can you actually go to a biobank, which has a much broader spectrum of people and different ascertainment? And we wanted to see, first of all, does it prove out that most of the variants with high odds ratios actually have disease? Great, that's how much do they line up with pathogenic or benign labels that existed before? But we also want to kind of calibrate that strength of evidence that we're talking about on the computational side. One of the things here to point out is that biobanks provide really large numbers of participants, and they collect these data from medical systems. Like the UK Biobank is just collecting ongoing clinical information, that's already going to exist in the medical system over time with lots of different phenotypes, lab measurements and things like that. So this is a really big strength. And observational data differs, based on this ascertainment and phenotyping. So you might say, oh gosh, FH, familial hypercholesterolemia, is something that's not always phenotyped in a patient. But we're going to think about, does a proxy for it exist? Can you calculate from LDL cholesterol levels whether or not there's a likely increase of hypercholesterolemia from having this variant? We can kind of create quantitative proxies. All right, I'm in the weeds here. Let me get out of the weeds. So one of the things I want to call out about this biobank data is that there's a really nice uniformity of data collection and variant processing. So when you think about resources like Nomad, they've gone through joint calling, they've thought about how to do that calling well, they've done the different sequencing techniques on the same platform. So it's really impressive in terms of thinking about that quality compared to kind of pulling case evidence from lots of different clinical labs. That part is really messy usually. So kind of amalgamating data from lots of different platforms gives you chances of false positives that don't exist here. So it's kind of nice. There are some pros of using this type of data. So what we did, we thought about, how do we take that framework of looking for cases and thinking about it only in the context of rare, highly penetrant, severe disorders? Can we kind of get it down to endophenotypes that are in everyday people, like cholesterol and hemoglobin A1C? And can we pull variants that exist in the genes that we already know about and see if they actually do increase or decrease risk of having those phenotypes or endophenotypes, for example, like lipid traits. And so let me just take a kind of step back at this. One thing I want to kind of call out here is a lot of folks would wonder, do you actually have enough patients with let me take hypertrophic cardiomyopathy or dilated cardiomyopathy? These are rare disorders. So if you think about a prevalence of a disorder of 1 in 500 people, pretty moderately rare disorder, still there are a lot of people in the UK Biobank with it. And so there might be ascertainments, questions, but still you're in the realm of having a lot of people with these disorders. And if you look at a variant that's even quite rare, 0.001%, you still have 5 individuals with it. So you still have some chance to make an epidemiological measurement. 1 person has disease, 4 people don't have disease, or 5 people have disease out of 5, right? So that gives you some sense of whether or not this variant is likely to cause disease in a bunch of people. And so I'm just calling out that there are a lot of folks that have various phenotypes in the UK Biobank, and that this work has, is building on prior work where we saw that endophenotypes that exist in population biobanks are predictive for deciding whether or not something is likely to cause disease using ClinVar data. Just racing ahead though, sorry, because I don't want to run out of time. So to do this work, we developed phenotype case definitions for 18 different phenotypes that are related to 41 genes in that ACMG secondary findings list, those actionable disease genes. And we used a variety of data from biobanks. We used self-reported data, hospitalization records, procedural codes, cancer registries, death registries, quantitative phenotypes, as I mentioned, like LDL cholesterol or hemoglobin A1C or long QT, or the QT interval in ECG. And we also did this work by gene and by phenotype when there were low numbers of ClinVar variants. Oh gosh, okay, I'm really in the weeds again. So let me think about this. So let's just take an example. LDLR is a gene that, if you break the LDL receptor, you're likely to have elevated LDL cholesterol. So what we did was we looked at the statin-adjusted LDL values, and we looked for patients that had an LDL cholesterol of over 190 milligrams per deciliter, and we counted that person, a person with that high level of LDL cholesterol, as a case. And we counted everybody else as a control or a person who was not a case. And same thing with breast and ovarian cancer. We made a case definition for that, colorectal cancer for everybody with a variant in MSH2. And we calculated the odds ratio for each variant, whether or not that variant had a high odds ratio of causing high cholesterol or breast cancer or colorectal cancer. And then we calibrated those. But I want to just show you, let's just go to some data, right? So if you look at every single point here, this is every single variant in LDLR that's rare. Every single point relates to just one variant. What we did was we calculated the odds ratio for that variant, that patients with this variant have an elevated cholesterol. They have cholesterol level of over 190. And so some patients, these are the pathogenic and likely pathogenic variants from ClinVar, these all do have quite high odds ratios in the UK Biobank. So there is this enrichment of patients with high LDL cholesterol for these pathogenic variants. Great, that perfectly makes sense, right? Like we already called this a pathogenic variant. There is this spectrum. I think it's worth noting that there are some of these variants that are not screaming awful variants that cause really high cholesterol in a lot of patients. So maybe these are less penetrant, or maybe they have a moderate effect. Maybe they're just statistical noise. But there is this great enrichment of odds ratios, high odds ratios causing high cholesterol for variants that we already labeled as PLP, pathogenic or likely pathogenic. Same thing with benign, kind of in the other direction. Benign and likely benign variants don't have an enrichment of high cholesterol patients in the UK Biobank. Then what we did was we took these VUS cases and we looked at the spectrum of different odds ratios, and we saw that there's this really high range of values. So there are patients with really high, these variants with really high enrichments of patients with high cholesterol, and other variants that have no enrichment at all, no apparent enrichment. And so what we did was we tried to, just like with those computational tools, we tried to calibrate the strength of evidence as that odds ratio goes up. Kind of, before we're talking about a score from a computational tool from 0 to 1, now we're looking at, an odds ratio value, a log 10 of an odds ratio value. And as this goes up, as the odds ratio goes up, we're more and more sure that this variant is likely to cause disease. So we have very strong evidence even for some very high odds ratios. And if you think about this is just like that blue and red dot point before. If you kind of combine these two columns, there is some range where you're pretty confident that this is going to be a pathogenic variant. And there's some range here where you're pretty confident this is going to be a benign variant. So this score range is now an odds ratio range. And so what we're able to do from this is basically calculate what those score thresholds are, which give you some confidence that this variant is likely to cause disease using population data. And so we kind of took a second here on cross-validation. We looked at whether this really works with a different phenotype. So we actually looked at whether or not people get heart attacks who have a score range of LDLR odds ratio variance of greater than 4.59. So somewhere around here, we looked at anyone with a variance value above that range versus anyone with a variant value below that range. And we found out that if you have one of these variants just with this type of evidence, there's no significant difference of having a heart attack from having a pathogenic variant that was already labeled as pathogenic in LDLR. So this is, there is some evidence that's coming from these population biobanks. Go for it.
It work. I'm just wondering, the cutoff that you use, if that's maybe not making use of all of the data because it's actually quantitative, right? So have you thought about doing that, or have you?
Yeah. So this is like a weird cutoff, right? This is in the paper we look at what is kind of the optimal cutoff. This is just one example, but of the clinical, this was to more toy with what is the right cutoff, right? What is the right cutoff that gives you this level of evidence? So this is sort of the question is like, can you calibrate it to come up with a cutoff? To kind of in the kind of clinical context of the way that people interpret variants, this is the kind of right answer. For this context, this is not like, I can't make a statement about this. It's just not significantly different, right? So I can't say that, oh, this is also causal, but I can show you that it's not so different than the pathogenic variants, but it is significantly different than having no variant, for example. But is the question sort of more like, what's the right way to draw the cutoffs, or?
Quantitative.
Oh, yes. Oh, that cutoff. Yes, 100%. So we tried 220. Yeah, so what we tried to do with these quantitative traits is we tried to use commonly applied clinical threshold labels. So like 190 is actually the pediatric level of what we would call familial hypercholesterolemia. 220, it might be an adult level. It may depend on sex. So there are different standards and certainties for this kind of levels that are commonly applied. Same thing for with like long QT, for example. We applied 2 different levels, one for men, one for women. But yeah, you can definitely move those. And I think that's a really valuable question because we tried to do this. We actually, that was a bigger part of this in the beginning in the story, thinking about like, what is the right cutoff? Because that gets to a different question, which is like, how do you know if you have a variant that has a moderate increased risk versus a very severe increased risk? And I think that's a good way to kind of tease that out. So I think it's a really insightful way to think about this. Yeah, any other questions? Okay, I am running tight on time, so I'm just gonna kind of tell the end of this story and say that we were able to do this for a whole bunch of genes individually. So we saw, we basically made these same plots for every single gene that we ended up developing data for. We're able to calibrate each gene differently. So some genes looked spectacular, like LDLR, but other genes like PCSK9, we know has the opposite effect for many variants. So it's like kind of nice. You don't get this same certainty. But what we did was we did this for lots of different variants and lots of different genes. And one of the things I want to kind of call out here, these are the colorectal cancer genes, the Lynch syndrome genes, but they're, we're able to kind of get these really nice separations between existing known pathogenic and benign labels for variant odds ratios of causing these same diseases in the UK Biobank, and also calibrate that level of strength of evidence for each of those different genes. Some of them were not very good because calibration is really hard when you don't have a lot of benign variants. There's a whole thing that is covered in that talk from 2024. And yeah, so the key findings were just like, we were able to kind of pull out for 2.6% of participants in the UK Biobank that they harbor a rare VUS. So this is a variant that doesn't have a classification with at least moderate evidence of pathogenicity, indicating a substantial disease risk that might be underappreciated and may be a new source of evidence that we can use in variant interpretation, like those computational scores. Maybe we have a new place, like a new way to kind of calibrate that strength of evidence from a biobank and draw that out as new information that can be used to classify variants. And so with that, I better call it, right? Because I think we're on time, or? Okay, cool, yeah. Let me think, did I have anything? Oh yeah, one other thing to point out is that this is, all of these strength levels are highly dependent on the choice of prior probability of disease. So if you're really, let me think about how to say this better, right? So if you're doing an adult monogenic diagnosis, you have a high prior probability. This patient came in because they they have a family history of disease. It's totally different than looking at population screening. So just thinking about how that influences when we were calculating posterior probabilities, they were on the basis of a prior probability of disease. So we're thinking about how much does this score push us over toward thinking this is a disease variant because it has a really high computational score. Well, that's still on the bed of whether or not that patient's likely to have disease given its, that patient's ascertainment. So this, we came up with a few different ways to do this that are our own in the context of biobank patients, but also there are commonly applied priors for like monogenic adult sequencing, for pediatric sequencing, et cetera. So there are all sorts of different ways to calculate those priors, and they're really influential for whether or not this variant or this score is likely to be strong evidence or moderate evidence. So this can really move these things. We tried to do this as honestly as possible, but I think, and they're very often very aligned in terms of the outcomes, but it's worth calling out. With that, thank you so much for great questions. And any others? I think we have a couple of minutes for questions, or?
Well, first, let's thank our speaker. Any quick questions in the room? I guess I'll ask, did you look at how these variant odds ratios replicated across biobanks?
No. So we didn't look at how they replicate across biobanks, and we didn't look at a couple of other factors. Like we didn't look at how these rules might contrast with the existing rules. So we're thinking about those things now. We're also thinking about, kind of along the lines of your question, thinking about how do you take these more common variants. So we exclusively worked on really like relatively rare variants with a frequency of 0.1% and below. But there is that range of variants that are between 0.1% and 1%, for example, that still have some disease risk that may be incompletely penetrant or may have a moderate effect size. We're thinking about both of those things right now. But we didn't look across biobanks, but we did cross-validation. So we ended up like looking at, we made predictions on one chunk of the cohort, and then we developed a model using one chunk of the cohort, like what are the odds ratio thresholds, for example, and then use those on other patients.
Great, that was fantastic.
Thank you.